Hello, I am Cecilia Mir and I will continue our talk about mutations. We learned about mutation in the previous video and now I will explain the causes, types of mutation and their effects. The main purpose is to identify and illustrate possible changes in nucleotide sequence in DNA and evaluate the significance of those changes in the resulting protein product. Okay, there are lots of different mutations that can occur in our DNA. Mutations can be classified in various ways depending on the cause of the mutations, its effect on the function of the gene product, or the kind of changes to the structure of the gene itself. The mutation that occurs suddenly in nature under natural conditions are called spontaneous mutation, like we will see. The origin is unknown. The mutation that can be artificially induced in the living organisms by exposing them to abnormal environments such as radiations, physical conditions, for example, temperature, and chemicals, are called induced mutations. The agents which induce artificial mutations are called mutagens or mutagenic agents. Scientists wanting to study mutations in a specific genome may induce mutations using a control lab setting, which will cause the number of mutations per unit of time or frequency to increase. Scientists induce mutations so they can study more effects in a shorter period of time. They induce these mutations by using a mutanizing agent such as radiation, chemical agents, or transposon insertion. A transposable element or transposon is a DNA sequence that can change its position within a genome, sometimes creating or reversing mutations and altering the cell's genetic identity and genome size. Sorry for my English. Okay. Yes. Genes and chromosomes can mutate in either somatic or germinal tissue, and the changes are called somatic mutations and germinal mutation, respectively. If a somatic mutation occurs in a single cell in developing somatic tissue, that cell is the progenitor of a population of identical mutant cells, all of which have descended from the cell that mutated. A population of identical cells derived asexually from one progenitor cell is called a clone. Germinal mutations occurs in the germline, special tissue that is set aside in the course of development to form sex cells. If a mutant sex cell participates in fertilization, then the mutation will be passed on to the next generation. Okay. Another way to classify mutations is depending on the extent of the material affected by the mutation. There are 3 types of mutations, 1 to 3, depending on the extent of the material affected. Mutation can affect anywhere from a single DNA building block, base pair, and we have the small scale mutation called gene mutation or point mutation, and we have the large scale mutations called chromosomal mutation and genomic mutation. We will see with more detail in the next slide. Okay, we will start talking about the point mutation that involves changes in one or few nucleotides. For example, substitution. Substitution mutations are situations where a single nucleotide is changed, for example here, GGTCT, GGTCA. Okay, a single nucleotide is changed into another, T changed to E. to A. In organisms having double-stranded DNA or RNA, this usually means that the corresponding base pair is also altered. For example, if we change this T in DNA for EA, will affect the codon, okay, GUG. This codon in RNA is the transcription chains and also the amino acid in the translation. If the amino acid change was really important for the structure and the function of the finished protein molecule, the simple fact of single base changes will cause the presence of a disease. This is the case of the sickle anemia. Okay. According to the effect caused in the final protein, we have different types of mutations by substitution of bases. The first point mutation, we can see here, is silent mutation, where no amino acid change. It's the same, lysine, because we change C to T, but it's a different codon, but codes for the same amino acid. Nonsense mutation is also a change in one DNA base pair, okay, here change A to U. Instead of substituting one amino acid for another, however, he altered DNA sequence prematurely signals the cells to stop building a protein. This type of mutation results in a shortened protein that may function improperly or not at all. Missense mutation here can be conservative or non-conservative. This type of mutation is a change in one DNA base, C, for example, T changed to C, that results in the substitution of one amino acid for another in the protein, okay, lysine changed for arginine, for example, in the protein made by a gene. If the properties of amino acid remains the same, it's called conservative. But if the amino acid has different properties than the wild type, for example, this has different properties than this one, the protein can lose its function, which can result in a disease in the organism. Okay. Another group of gene mutation is called frame shift mutation. This type of mutation occurs when the addition, okay, insertion or loss of DNA bases, deletion, changes the gene's reading frame. Remember that a reading frame consists of groups of 3 bases that each code for one amino acid. A frame shift mutation shifts the grouping of these bases and changes the code for amino acids. The resulting protein is usually non-functional. This way, insertion, deletions can be all frame shift mutation. As we can see in this image, deletion changes the number of DNA bases by removing a piece of DNA. Small deletions may remove one or a few base pairs within a gene changing a gene's reading frame. In this case, it's the removal of one simple nucleotide, for example, okay, that changes all codons. Okay, it is the original, the wild type. If you change only one, if you lose one simple nucleotide, okay, all these codons are different. The same occurs with insertion, but in this case it's because of adding DNA bases. Okay, so frame shift mutation are insertions, the deletions in the genome that are not in multiples of 3 nucleotides. Because if there are 3 that change, for example, that change 3 nucleotides, it only changes one codon or one amino acid, but not all the codons, okay, after the mutation. Frame shift mutations are among the most damaging changes to the coding sequence of a protein. They're extremely likely to lead to large-scale changes to polypeptide length and chemical composition, resulting in a non-functional protein that often disrupts the biochemical processes of a cell. Frame shift mutations can lead to a premature end to translation of the mRNA, as well as the formation of an extended polypeptide. You can see, for example, the original message can be Sam and Tom ate the ham. Here it's nonsense message. The amino acid sequences downstream of the frame shift mutation are also likely to be chemically distinct, okay, from the original sequence. Diseases caused by frame shift mutation in genes includes Crohn's disease, phenylketonuria, cystic fibrosis, and some forms of cancer. Another type of mutation, depending on the extent of the material affected by the mutation, is called chromosomal mutation. Deletion, duplication, inversion, and translocation are the 4 types of chromosomal mutation. Deletion is when genetic material on the chromosome is lost. If a significant amount of genetic information is lost, the mutation is likely lethal. Duplication occurs when a portion of the chromosome is copied and replicated so that there is extra information. Inversion, in inversion, the order of some genetic material is reversed or switched. Translocation occurs when a piece of one chromosome breaks off and is transferred to a new location on another chromosome. Here we can see better. Deletion is when genetic deletion, here, is when genetic material on the chromosome is lost. Okay. A few nucleotides or large number could be lost. Deletions can happen anywhere on the chromosome. In this image, B, okay, has been lost. Duplication, this one occurs when a portion of the chromosome is copied and replicated so that there is an extra information. For example, here B has been duplicated. In inversion, the order of some of the genetic material is reversed or switched. A segment of the chromosome is reversed. For example, here no genetic information is lost, but if the break reversal occurs at a crucial point, then there can be serious consequences and the inversion could be lethal. In the image, for example, the inversion, okay, if, for example, A, it needs to be adjacent to B for any reasons, for some reasons, for the organism to survive, for example, the inversion will be lethal. Translocation occurs when a piece of one chromosome breaks off and it's transferred to a new location on another chromosome. In this image, the chromosome that originally began with ABC has lost the F, okay, DEF, and gained JKL. The F has been translated to the second chromosome, for example. Several forms of cancer are caused by translocation, for example. Finally, we're going to talk about the genomic mutation, another large-scale mutation. Okay, this genomic mutation. Okay. Is the fact that a large proportion of genetically determined ill health in humans is caused by abnormal chromosome numbers. Additionally, manipulation of chromosome number is routinely used by breeders to improve agriculturally an important species. Changes in chromosome number are of 2 basic types. Changes in whole, here, eplody, changes in the whole chromosome set, resulting in a condition of aberrant eplody, and changes in parts of chromosome sets, resulting in aneuploidy. The number of chromosomes in a basic set is called the monoploid number, X. Organisms with multiples of the monoploid number of chromosomes are called diploid. Eukaryotes normally carry either one chromosome set, haploids, or two sets, diploids. Haploids, for example, what is this? Okay, haploids and diploids then are both cases of normal diploidy. Okay. Diploid types that have more than two sets of chromosomes are called polyploid. Polyploids arise naturally as spontaneous chromosomal mutations, and as such they must be considered aberrations because they differ from the previous norm. However, many species of plants and animals have clearly arising through polyploidy. So, evidently, evolution can take advantage of polyploid when it arises. The polyploid types are, for example, diploid, tetraploid, pentaploid, hexaploid, and so forth. Aneuploidy is the presence of a normal number of chromosomes in a cell. Okay, for example, a human cell having 45 or 47 chromosomes instead of the usual 46. An extra missing chromosome is a common cause of genetic disorders, including some human birth defects. Okay, here. Aneuploidy is the second major category of chromosome aberrations in which chromosome number is abnormal. Aneuploid is an individual organism whose chromosome number differs from the wild type by part of a chromosome set. Generally, the aneuploid chromosome set differs from a wild type by only one chromosome or by a small number of chromosomes. For example, here, aneuploids can have, aneuploids is nearly always deleterious because of genetic imbalance. The ratio of genes is different from that in euploids, and this difference interferes with the normal function of the genome. Okay? Aneuploids, for example, can have chromosome number either greater or smaller than that of the wild type. Aneuploids nomenclature is based on the number of copies of the specific chromosome in the aneuploid state. For example, if only one copy of some specific chromosome is present instead of the usual two found in its diploid progenitor, it's called monosomic. It's, for example, the Turner syndrome. In the Turner syndromes, we have a human female phenotype. In these syndromes, we have a human female phenotype produced by a presence of only one X. Here, it's only one chromosome, X chromosome, XO. Okay. There is also the trisomic condition. Also, is one of the chromosomal imbalance that can result in a normality of death, called Down syndrome. The combined phenotype that makes up Down syndrome include mental retardation, broad flat face, eyes with a chondric fold, short stature, short hands with a crease across the middle, and a large wrinkled tongue. Okay, you can see that trisomic is 3 chromosomes instead of 2. The only other human autosomal trisomics to survive to birth are trisomy 13, okay, it's called Pato syndrome, and trisomy 18, Edwards syndrome. Both show several physical and mental abnormalities. The phenotypic syndrome of trisomy 13 includes a hair lip, a small malformed head, rocket bottom feet, and a mean life expectancy of 130 days. That of trisomy 18 includes fawn-like ears, a small jaw, a narrow pelvis, and rocker-bottom feet. Almost all babies with trisomy 18 die within the first few weeks after birth. Other trisomies die in utero normally. Okay. Now to finish, I'm going to explain a metabolic disorder due to a genetic mutation in DNA sequence that I studied a few years ago. Concretely, it is a disease caused by a missense mutations. Remember that a missense mutation is a point mutation where substitution of one nucleotide for another causes a change of amino acid in the result protein. To know how important this change, you can see here's missense mutation, one amino acid changes to another. To know how important the change of amino acid can be for the protein, let's assume it is an enzymatic protein, for example. It is important to study first the function and structure of the protein. Second, it's important to see where the amino acid is located and its responsibility in this protein. And third, and no less important, it will be necessary to do studies of the enzymatic activity in the metabolic pathway to see if its activity is minor or even if it's null. Sometimes a single amino acid change causes a decrease of the enzymatic activity. When enzyme in a metabolic path, for example, in this case, we were talking about ketogenesis, or the same is synthesis of ketone bodies. So if an enzyme of this pathway stops working, or it works worse, for example, human lyase, yeah, two things can happen. If this enzyme works worse, or stops working, two things can happen. One thing, accumulation of its substrate, in this case, this one, causing toxicity in the organisms. And another thing that can happen is lack of products. In this case, these three products or three ketone bodies. These three ketone bodies produce are important resources of fuel and energy for many tissues, including brain, heart, and skeletal muscle. When this happens, due to this toxicity, the person presents acute episodes that include vomiting, lethargy, hypotony, apnea, and sometimes evolves to coma. This is diagnosed by a 3-hydroxy-3-methylglutaric aciduria, a rare autosomal recessive metabolic disorder that appears in the first year of life. As we have seen, the origin of this disease, aciduria, is a mutation in the gene coding for 3-hydroxy-3-methylglutaryl coenzyme I, okay, also called human lyase, HL, enzyme. This enzyme, okay, human lyase enzyme. This enzyme cleaves this substrate to form these products, acetyl-CoA and acetoacetate. Acetoacetate. Oh my goodness, it's very difficult in English. Okay, we propose a Tim-Burrell folding for the enzyme, human lyase. The Tim-Burrell is a highly conserved protein, a structural domain consisting of 8 alpha helices and 8 parallel beta sheets that alternate in the protein skeleton. Okay, you can see the Tim-Burrell here. Okay. In the proposed model from human lyase enzyme, the side chains of the barrel form beta sheets that conform the central activity, these beta sheets, okay, called enzyme active site. This is the active site. where the substrate is assumed to be accommodated. Okay, this is the cavity, okay, where the substrate here, okay, is assumed to be accommodated. Okay. When the original amino acid, these both are original amino acids, are replaced by others, these others, okay, the proposed substrate cavity here is the substrate cavity. In the mutated protein, this cavity is occluded by the longer side chain of the mutated amino acid, and the entrance of the substrate is prevented, which is consistent with the complete absence of the activity of certain expression studies performed with mutants. Okay, you can see here the absence of activity. Okay, because the cavity is occluded. Well, that's all for now, and don't forget, the roots of education are bitter, but the fruit is sweet. Thank you for your attention and see you soon. Bye.